Hello everyone, I'm Colette McKay from the Biomedicine Institute. I'm the principal scientist there and I lead the translational hearing research team. So I'm an auditory scientist, so my aim in life is to improve the lives of people with hearing impairment. And I do this by applying a combination of different techniques through basic science right through into translation into clinics. So I'm going to take you on my journey now. First of all, I'm going to just tell you my geographic journey about my career. So I started off doing a PhD in physics at the University of Melbourne. And following that, I did a graduate diploma in audiology. And from there, I took on research fellowships to work on cochlear implants. And during that time, I had exchange visits with Cambridge University in the UK. So later, I took up a chair in auditory science position at Aston University in Birmingham in the UK. And from there, I went even further north to the University of Manchester, where I led the audiology research group there. And finally, I came back to Melbourne with a Vesky Innovation Fellowship for the Biomedicine Institute, where I'm currently the principal scientist. Now that's my geographic journey. Now I'll tell you about my science journey. So my very first research quest was in the 1990s. How can I help profoundly deaf people hear again using cochlear implants? And how can we progress from the very early stage version that was basically only an aid to lip reading? So people with cochlear implants wanted to use a telephone. They wanted to give up lip reading and just use their implant to hear sounds. So how did we do this? Well, I teamed up with a clever engineer, Hugh McDermott, who developed a good idea for a better way to code acoustic information into the electric signal. So this better way was called the Spectral Maxima Sound Processor, or SMSP. And we teamed up to show how well it worked. So I'll show you something about the SMSP. So on the left here, we have a spectrogram of the word wish. So the vertical axis is frequency and the horizontal axis is time. And you can see that as you say wish, you can see the low frequency vowel sounds and the transition where you go ooey, and the shh sound here at the higher frequencies. Now, the old signal processing strategy did this. So this is very similar to here. Instead of frequency, we've got electrodes. So these electrodes are tone-topically arranged to match certain frequency bands. And we've got time on the bottom again. And each little bar is an electrical pulse. And you can see the pattern is the same as over here, but it's very sparse. This is the new one here, and you can see the information is a lot less dense, and particularly in this rapid transition here where you go, we got a lot more information for the person to hear. And so this signal processing strategy was commercialised by Cochlear Limited as the sound strategy called Speak, and it's the most it's the basis of the strategy that's used today in cochlear implants still. This is just a graph showing that the Speak strategy turned out to be much better than the current one at the time. So that was my first episode, if you like, in my research career. So how did I go from there for the last 20 to 30 years improving cochlear implants even further? In the middle here, you can see all the different types of science that I've had to use to actually do further improvements to cochlear implants. Psychophysics is the measurement of perception. Speech processing studies, signal processing, which is about converting the information in an acoustic signal into electrical stimulus patterns. Electrophysiology, which is the measurement of actual neural responses, the electrical signals that the nerves create when they fire. and brain imaging techniques. So these red circles are the bits of basic knowledge that come out of those studies. And so this is sort of the basic science part of it all. So we could find out what was the relationship between the parameters of electrical stimulus and what people heard. How does the auditory, the auditory parts of the brain actually work? What's the difference if you actually stimulate higher regions in the brain instead of the cochlea? And how does deafness change the brain? So doing basic research and finding out all of these things led to actual translational outcomes. So we're developing automatic programming of cochlear implants for infants. Of course, infants can't tell you how much they can hear and whether it's too loud or not. Improved signal processing strategies and individual optimization of benefit for patients. So I'm going to show you some example of that pathway. So this is a pathway based upon the perceptive loudness. So loudness is different from level. Okay, level is what you can measure with a sound level meter. Loudness is what people perceive as the loudness of a sound. So with psychophysics, we determined how do parameters of electrical stimulus affect the loudness perceived, and how does that differ from analogous effects in acoustic stimulation. And from there, we derived a mathematical model to describe the auditory processing from cochlea through the central processing system to perception that governs loudness. So on the left, we have the output of a loudness model, and on the right, you can see that the predicted loudness and the measured loudness are actually very correlated based upon the model. So where did we go from there? We used electrophysiology to validate the model. And so this is a graph of electrical evoked compound action potential. So that's the whole auditory nerve response to single pulses. And that's the amplitude of it. And these are the amplitudes to different pulses in pulse trains with different rates of stimulation. So after that, we get to translational benefits. So from that loudness model, we can do improved automatic and objective programming of cochlear implants. We developed new patented signal processing strategies, and it also helped to develop a new auditory prosthesis that electrically activates neurons in the brain. So I'm going to actually show you this last example here. So what's a midbrain implant? This is a picture, a schematic of the auditory pathways from the cochlea to the brain. So a cochlear implant has electrodes that go in the cochlea, of course. And there's another type of implant called an auditory brainstem implant, where electrodes are put on this first brainstem part here called the cochlear nucleus. Now, auditory brainstem implants don't work very well. They don't work nearly as well as cochlear implants, but some people need those because their cochlea is not suitable to actually put a cochlear implant in. And so these people actually need to have some sort of prosthesis, but it doesn't work very well. So along comes the auditory midbrain implant, which has a shank of electrodes that go into the inferior colliculus even higher up here. So the first version of the auditory midbrain implant didn't work any better than the brainstem implant. This is the old design where you've got a single shank of electrodes going in all the different frequency layers here in the inferior colliculus. So what we did is we took those patients and we did psychophysics again to understand why they didn't actually get the benefit we were anticipating. And we produced a hypothesis for why that was so from those studies. Then we did an animal electrophysiology to actually validate the hypothesis. And this resulted in a new AMI design where we have two different shanks, and so you can actually stimulate different areas within the same frequency lamina here. And this new design is now in clinical trial. That's just one example of going from basic science through to translation. So finally, I just want you to, I want to tell you about my latest big experiments at the Biomedicine Institute, where we're actually trying to improve objective assessment of infants. So infants who are picked up by newborn hearing screening go through a diagnostic process, and it's really important that they actually, a hearing impaired infant actually has access to sound straight away. So they need to be fitted with a cochlear implant or a hearing aid so that they can access sounds and so they can develop oral language. This is really important. So what are we doing now to actually improve this situation? We're using a technique called functional near-infrared spectroscopy, or fNIRS, to measure the discrimination of speech sounds. So fNIRS uses near-infrared light to actually image brain activity. It's the same signal that fMRI uses, which is the blood oxygen changes in the brain when different parts of the brain become more active. But instead of having a big fMRI machine, what you've got is just a little cap that fits on the baby's head, and it has little light sources and light detectors. Using this technique, we've shown that we're able to actually show that an infant can distinguish between different sounds. So here's the fNIRS amplitude of a bar sound and a T sound. They're very different. So we can see that the brain of the baby can distinguish these things. So this is leading to a new translational development of a new fNIRS device for audiology clinics. So that's a very brief rundown of my research career. I hope you enjoyed listening. Thanks very much.